The overall goal of this procedure is to inoculate an aerobic wood-degrading fungus in a petri dish microcosm where wood moisture and exogenous substrates are tightly controlled. This is accomplished by first preparing microcosms with exact volumes of known agar media. Then, the wood to be degraded and any exogenous solid substrates are assembled on top of a plastic grid on the agar surface without allowing contact among substrates. The microcosm is then inoculated and incubated with the test fungus. Following incubation, wood blocks are harvested to determine mass loss and to prepare tissues for analyses. Ultimately, results can be obtained that show the degree of wood biodegradation and the role of exogenous element sources through mass loss measurements, observations of mycelial morphology, and targeted elemental analyses like inductively coupled plasma spectroscopy.
Hi, my name is Brook Jacobson from the laboratory of Dr. Jonathan Schilling in the Department of Bioproducts and Biosystems Engineering at the University of Minnesota. Today we're going to show you a procedure for using petri dishes as microenvironments for the plant tissue decomposition by aerobic fungi. We use this procedure in our lab to study the mechanisms of fungal lignocellulose decomposition. Lignocellulose is the plant tissue comprised of lignin, cellulose, and hemicellulose. The tissue we study most often is wood. Mistakes with the less technical aspects of Agar-Block design are the most common causes of failure and the key sources of variability among studies, making video publication of our Agar-Block setup useful. We know from experience that this procedure yields high-quality, low-variability results. So, let's get started.
For these experiments, use petri dishes that are 1 centimeter deeper than typical petri dishes to increase the headspace above the wood block in the microcosm.